If I were to ask you to define immortality, you'd probably start thinking about, vampires or maybe some futuristic sci-fi thing where you upload your consciousness to the cloud.
Right. Living forever, bypassing the end of the line.
Exactly. But in the world of biology, um, immortality is a very real, very specific technical term.
Hmm.
And it doesn't look like a vampire. It looks like a microscopic sample of tissue that just won't stop dividing.
That's absolutely right. And for decades, scientists were chasing this biological holy grail. They wanted human cells that wouldn't just die in a petri dish after a couple of days.
And you have a stable supply.
They needed something that could survive long enough to actually be studied, to be tested and experimented on. It was the primary bottleneck of medical research in the early 20th century.
And they finally found it in 1951. But here is the paradox that we're really going to unpack today.
This is a huge one.
If you could somehow gather every single one of these immortal cells,
Yeah.
every microscopic sample currently living in a freezer in Tokyo, a petri dish in London, or, a biofactory in California, and you piled them all into a scale,
Yeah.
they would weigh tons, literal tons.
Which is it's mind-bending when you consider the origin of it all.
Exactly, because we aren't talking about a fungus or some kind of synthetic material. We are talking about human tissue, biological matter that originated from one single woman.
A woman who stood about 5 feet tall, weighed maybe 150 pounds, and died more than 70 years ago.
It's a story of a massive scientific triumph that is just inextricably bound to a profound human tragedy.
We're talking about the HeLa cell line. That's HeLa.
Which, as we'll find out, is just a code name. The first two letters of a first name and the first two letters of a last name. For a long, long time, people thought it stood for Helen Lane or
Helen Larson. That was another common theory.
But it didn't. It stood for Henrietta Lacks.
Precisely. And the mission for this deep dive is to bridge that gap. We have the HeLa cells, this ubiquitous, immortal tool that was used to cure polio, to map the human genome.
All the greatest hits of modern medicine.
And then we have Henrietta Lacks, the African-American tobacco farmer and mother of 5 who lived in poverty and died in pain, completely, totally unaware that she was about to become the mother of modern medicine.
We're going to look at the science, sure, but we really need to get into the ethics, the theft, if we can even call it that, and the family that was left behind. Because for 25 years, her own family didn't even know these cells existed, right?
They had absolutely no idea. While Henrietta's cells were being, bought and sold by the vial, her family couldn't afford health insurance.
Wow.
That is the core tension we are navigating today. It's this collision of poverty, race, and what we call scientific progress.
So let's get away from the microscope for a second and actually meet the woman. Who was Henrietta Lacks before she became this medical acronym?
Well, she was born Loretta Pleasant.
Loretta?
Yes. Born Loretta Pleasant on August 1st, 1920 in Roanoke, Virginia. The family actually isn't sure how or why she switched from Loretta to Henrietta, though she was always nicknamed Henny. But her start in life was incredibly difficult. Her mother died in childbirth when Henrietta was just 4 years old.
4?
Giving birth to her 10th child.
Wow. 10 kids. Losing your mother at 4. That's a hard life from day one.
It was overwhelmingly difficult. And after her mother died, her father he couldn't handle all the children. He didn't have the resources or the capacity. So, as was often the custom in that community at the time, the kids were distributed among relatives. Split up. Henrietta was sent to Clover, Virginia to live with her maternal grandfather, Tommy Lacks. And the setting here is really, really important for understanding the history. They lived in a 2-story log cabin.
Okay, I'm picturing a log cabin, rustic, rural Virginia.
More than just rustic. This cabin was formerly the slave quarters of the plantation that had been owned by Henrietta's white great-grandfather.
Wait, say that again?
She was raised in the same cabin that was used as slave quarters on the plantation owned by her own white ancestor.
So she was literally growing up in the shadow of slavery, living in the very building where her ancestors had been enslaved.
Exactly. It puts the timeline in perspective. We think of this as modern medicine, 1951, but the legacy of the plantation is literally the roof over her head.
That's incredible.
And her daily life was defined by hard labor. From a very young age, she was a tobacco farmer. She was out there feeding animals, tending gardens, working the fields from dawn until dusk.
What about school?
She did attend the designated black school in the area, but she had to drop out in the 6th grade.
6th grade?
To work full-time to support the family. There was no other choice.
So she has a 6th grade education, she's working tobacco fields and living with her grandfather, And this is where she meets her husband, right?
Yes. It's another part of the story that feels from a different time. She was actually sharing a room with her first cousin, David Lacks. They grew up together in that cabin. They eventually married in 1941. Henrietta was just 20, but she had actually already given birth to their first son, Lawrence, when she was only 14.
14. It's
Yeah.
just, it's a different world.
It was. And eventually, they did what millions of African Americans did during that era. They joined the Great Migration. They left the tobacco fields of Virginia to seek better industrial jobs up north.
Because the farm life just wasn't sustainable.
It offered no way out of poverty.
Yeah.
So in their case, they moved to Turner Station, which is near Dundalk, Maryland.
Right outside Baltimore.
Exactly. Day got a job at Bethlehem Steel at Sparrows Point. It was a huge shipyard and steel mill. It was grueling, dangerous work, but it paid better than tobacco.
And a family got a house of their own.
They did. With some savings gifted by a cousin who was going off to war, they bought a house at 713 New Pittsburgh Avenue. And they had 5 children in total. Lawrence, Elsie, David Jr., who they called Sonny, Deborah, and Joseph.
I want to pause on one of the children you just mentioned, Elsie. Because in reading through the source material, the story of Elsie seems like this dark, tragic foreshadowing of what happens to Henrietta.
It really does.
It feels like an anchor on the family's history.
It's just heartbreaking. Elsie was their second child, born in 1939, and she had, well, what we would now call epilepsy and cerebral palsy. At the time, the family just described her as different or deaf and dumb,
They didn't have the medical language for it.
or the resources to help her. And so eventually she was institutionalized at a place called the Hospital for the Negro Insane.
Just the name alone sends a shiver down your spine.
That was its official name. It was later renamed Crownsville Hospital Center. But the reality was, well, it was worse than the name.
What happened to her there?
Elsie died there in 1955 at the age of 15. But years later, when historians and Rebecca Skloot looked into the practices at Crownsville, they found it's highly possible Elsie was subjected to a procedure called pneumoencephalography.
I am almost afraid to ask, but break that down for me. Pneumoencephalography.
It's a procedure that is considered absolutely barbaric by modern standards. They would drill holes into a patient's skull
Oh my God.
to drain the cerebrospinal fluid, the fluid that cushions the brain. Then, to keep the brain's structure from collapsing and to create contrast for imaging, they would pump air or helium directly into the skull
Why would they do that? For an X-ray?
to make the brain stand out more clearly on an X-ray film. The fluid usually makes the image cloudy, so this was their solution.
What were the side effects?
Horrific.
Right.
Excruciating headaches that could last for months, vomiting, seizures. It was effectively torture, and it was often performed on patients who could not consent, who couldn't even tell you what was happening.
That is horrifying. And Elsie was just a child.
She was, and this is the context. This is the Lacks family's interaction with the medical establishment before Henrietta ever got sick. It's a history of poverty, of lack of agency, and of institutions that viewed them as subjects for research rather than as patients to be cared for.
So we have Henrietta in Maryland. It's now 1951. She's 30 years old. She's just had her 5th child, Joseph. But she knows something isn't right.
She did. She felt what she called a knot in her womb. She had actually mentioned it to her cousins before Joseph was born, thinking maybe it was just the pregnancy, but after the birth, the knot was still there.
And then it got worse.
Then the hemorrhaging started, severe bleeding.
So she goes to the doctor, and she ends up at Johns Hopkins. Why there specifically?
Because it was 1951 in a segregated city. Johns Hopkins was one of the very few top-tier hospitals that would treat African-American patients in its public wards, the so-called colored wards.
She didn't really have a choice.
Not for the kind of care she needed, no.
And she sees a doctor there named Howard Jones.
Correct. Dr. Jones examined her and found a mass on her cervix. It was described as shiny and purple. A biopsy quickly revealed it was a malignant epidermoid carcinoma.
Cervical cancer?
Yes. Now, there's an interesting side note here. Years later, in 1970, doctors re-examined her tissue slides and realized she had been misdiagnosed. It was actually an adenocarcinoma.
Okay, would that have changed her treatment back then?
Probably not. The standard treatment at the time for cervical cancer was radium, regardless of the type. However, adenocarcinoma is much, much more aggressive than epidermoid carcinoma. It helps explain why the cancer spread so incredibly rapidly and, why the cells themselves turn out to be so unbelievably robust.
So let's talk about that treatment. You mentioned radium. How did that work?
They would insert these tubes of radium, a highly radioactive element, directly against the cervix to burn the cancer cells away. It involved sewing pouches filled with this material right inside the patient's body.
Which sounds incredibly painful and dangerous by today's standards.
It was highly invasive and harsh. It caused the skin to blacken and burn. But here is the pivot point of history. This is the moment where Henrietta Lacks becomes HeLa.
During the procedure,
During her treatment, while she is unconscious on the operating table, a surgeon takes 2 samples from her cervix.
without her asking.
Without her asking, without her permission, and completely without her knowledge. 1 sample was of her healthy tissue, and 1 sample was from the cancerous tumor.
Okay, let's unpack this without permission part. Because looking at it from today's perspective, this just screams lawsuit. It screams violation. But what was the context in 1951?
And that's the nuance we have to understand to get the full story. In 1951, the concept of informed consent, as we know it today, it just didn't exist.
It wasn't a law.
It wasn't required by law, and it wasn't customarily sought by doctors, especially not in the public wards of a hospital. The prevailing attitude was that if you were receiving free or subsidized care, the price you paid was being available for research.
So your body was sort of talk hospital property while you were there.
In a way, yes. The patient was unwitting, absolutely. But strictly speaking, the doctors weren't breaking a law. They were following the norms of a deeply paternalistic medical system.
Which doesn't make it right, of course.
Not at all.
Yeah.
But it explains how it happened so casually.
Mhm.
It was just standard operating procedure.
So they take these 2 little samples. What happens to Henrietta?
The treatment failed.
Hmm.
The cancer was just incredibly aggressive. It metastasized throughout her entire body. By August 8, 1951, she was in severe pain and was admitted to the hospital for the last time.
And she died not long after.
She died on October 4th, 1951. She's only 31 years old.
And she's buried in an unmarked grave back in Clover, Virginia, right?
Yes. And the family cemetery at a part of town called Lackstown. For decades, her grave had no stone. She just disappeared into the earth.
While her cells were about to take over the entire world, so let's move from the operating room to the lab. Who was on the receiving end of those samples taken at Hopkins?
That would be Dr. George Otto Gay. He was a very prominent cancer researcher at Hopkins, and he had been trying for practically 30 years to grow human cells in culture, and he was failing.
Everybody was failing.
Everybody was failing. It was the biggest problem in the field.
What was happening to the cells usually?
They would put human cells in a nutrient medium, kind of soup made of chicken plasma and salts and things, and the cells would just die in a few days. You can't run long-term experiments on something that dies over the weekend.
So Gay gets Henrietta's cells. His assistant labels them HeLa. First 2 letters of Henrietta, first 2 of Lacks, and then what?
And then a miracle happens. His lab assistant, a woman named Mary Kubitschek, plated the cells, and unlike every single other sample they had ever seen, these cells didn't die.
They just survived.
Not only did they survive, they grew with what was described as terrifying speed. They were doubling their population every 24 hours.
Oh, why? What on earth made them so special?
It was this perfect storm of that specific super-aggressive adenocarcinoma combined with something in Henrietta's own biology. They didn't have cellular senescence, they didn't age, they just kept dividing. They were the first immortal human cell line.
The first ever.
Now, I should clarify. There were mouse cells that had been immortalized earlier, specifically a line called L929. But for human research, HeLa was the absolute breakthrough.
And Gay must have realized he had struck gold.
Oh, he did. He isolated one specific cell, divided it, and essentially created a pure, robust lineage that was strong enough to be shipped. You could put these cells in a test tube, mail them to a lab in Texas or India, and they'd survive the trip and start growing again as soon as they arrived.
I read about this roller tube technique they used to grow them. It sounded like something out of a factory.
It really was. Imagine a large metal drum, basically a cylinder with holes in it, and it's rotating slowly like a cement mixer.
Okay.
Inside the holes are test tubes containing the cells and the nutrient fluids. As the drum turned about once every hour, the fluid would wash over the cells to feed them, and then it would drain away so they could breathe. This constant, gentle motion allowed for mass production on a scale they had never dreamed of.
Mass production of human material, it's just wild. So suddenly, scientists everywhere have access to human cells that don't die. What does this actually unlock? What's the why it matters part of the story?
The impact is, it's almost hard to overstate. The first major victory was polio.
Right.
It's 1954. Jonas Salk is developing his vaccine. But to test it, He needs millions and millions of cells to see if the vaccine effectively neutralizes the polio virus without killing the cells themselves.
And he couldn't use monkey cells for everything?
Exactly. It was too expensive, too inconsistent, and ethically fraught. HeLa cells were the answer. They built a literal cell production factory at the Tuskegee Institute just for this purpose.
A factory for cells.
Yes,
Yeah.
but it didn't stop with polio. This is where we get into the broad utility. They used HeLa cells to study the effects of radiation. You have to remember, this is the dawn of the atomic age and the space race.
So they were testing the effects of atomic bombs on human tissue.
On HeLa cells, yes. And they sent HeLa cells up in the very first space missions to see what zero gravity and cosmic rays would do to human biology.
They sent Henrietta to space?
In a way, yes. Her cells went to space. They were used for gene mapping, which was revolutionary. They were used to understand how viruses like HPV and HIV work. They were even used for much more mundane things, like testing sensitivity to glue, cosmetics, and tape.
So if you've ever bought a hypoallergenic shampoo,
There's a very good chance that HeLa cells were involved somewhere in the long history of testing those ingredients for safety.
and they were used for cloning too.
Yes, scientists at the University of Colorado successfully cloned HeLa cells in 1955, the first human cells to ever be cloned. And today, there are over 11,000 patents that involve HeLa cells.
11,000.
But then there's the dark side of the research, too. You mentioned Chester Southam earlier.
Right. This is the stuff that makes you cringe reading about it today.
Chester Southam was a virologist who had a burning question. Was cancer contagious? Or could you build an immunity to it? So he started injecting live HeLa cancer cells directly into patients.
Into people who didn't have cancer.
Well, he started with cancer patients to see if their bodies, already weakened, would reject the new cancer. But then he moved on to healthy prison inmates at the Ohio State Penitentiary.
He injected prisoners with cancer cells.
He did. He told them he was testing for cancer, not that he was injecting it.
That's horrifying.
It raises massive ethical red flags now, but at the time it was viewed by some as cutting-edge research. It actually eventually led to much stricter regulations on human experimentation, but it happened. And it happened because HeLa cells were so readily available and so robust.
And all of this is happening. Salk and the polio vaccine, space travel, these horrifying prison experiments. And the Lacks family is doing what? Where are they in all this?
They're living in Baltimore, working, raising kids, struggling to get by. Henrietta died in 1951. The family knew absolutely nothing about her cells until 1975.
24 years. A 24-year gap.
A whole generation.
How did they finally find out? Did someone from Hopkins call them up and say, hey, by the way, your mom is a scientific superstar?
Not exactly. And the reason they were finally contacted is actually quite ironic.
How so?
By the 1970s, HeLa cells were, in a way, too successful. They were so aggressive and they traveled so easily on dust particles or unwashed hands that they were contaminating other cell cultures in labs all over the world.
Like a weed.
A scientific weed, exactly. Researchers would think they were studying breast cancer cells or prostate cells, but oops, it was actually HeLa that had invaded the petri dish and taken over. It was a huge problem.
So the immortality became a liability?
In that context, yes. So researchers needed to find a way to genetically identify HeLa to differentiate it from other cell lines. They needed the original DNA map. But Henrietta was dead. So who holds the closest genetic match?
Her kids.
Her kids. So researchers started calling the Lacks family, asking for blood samples. But here's the tragedy of the communication gap. The Lacks family had limited scientific literacy. They weren't explained the concept of cell culture contamination. When doctors from Hopkins called asking for blood,
They thought, what?
they thought they were being tested for the cancer that killed Henrietta.
Oh, man. So they're terrified. They're thinking, Mom died this horrible cancer, and now they want to check us for it.
Precisely. They were confused and alarmed. Henrietta's daughter, Deborah, lived in constant fear that she had the cancer and was going to die at 31, just like her mother.
So they still didn't even know the cells existed.
Not yet. It wasn't until a chance encounter in 1975 that the truth finally came out. A family member was at a dinner party and met a cancer researcher. When they mentioned their last name was Lacks, the researcher said, "Oh, like the cells. HeLa cells. They're famous. They're still alive."
Imagine hearing that at a dinner party. Your dead mother is alive in a lab.
It was a profound shock. And it wasn't a happy discovery. It was terrifying for them. They imagined her whole body was somewhere being experimented on, or that she was being cloned over and over. It just opened up this Pandora's box of confusion and pain.
And violation of privacy, too. Because once that connection was made, The family's medical history basically became public domain.
It got worse. In the 1980s, without consent, some of their family medical records were published in scientific articles. But the biggest breach came much more recently, in 2013.
2013.
Yes, scientists at the European Molecular Biology Laboratory sequenced the full genome of a HeLa strain and published it online for anyone to see.
And if you sequence Henrietta's genome, you are, by definition, partially sequencing her children and her grandchildren.
Exactly. JLX Way, Henrietta's granddaughter, put it perfectly. She said this wasn't just Henrietta's data. It revealed genetic traits, predispositions to diseases, all this hereditary information of the living family members. It was laid bare for any insurance company or employer to potentially see.
And how did they find out? Did the scientists call them?
They found out about this publication from Rebecca Skloot, the author of the book, not from the scientists themselves.
So 2013, that's practically yesterday. Did that public outcry finally force a change?
It did. The NIH, the National Institutes of Health, finally sat down with the Lacks family. They reached an agreement.
What was the agreement?
Now, access to the full HeLa genome is regulated. There's a committee, the HeLa Genome Data Access Working Group, and 2 members of the Lacks family sit on that committee. They help decide who gets access to the data and for what purpose.
So they have some control now.
Some. And researchers who use the data are now required to acknowledge Henrietta Lacks and her family in their papers.
But they don't get money, right? No financial compensation.
Under that 2013 NIH agreement, no, they do not receive financial proceeds from commercial products that are developed using the genome data.
And that leads us right into the legal and ethical battlefield, because I think most listeners right now are thinking, wait a second, pharmaceutical companies have made billions off these cells. The family was poor. How on earth is that legal?
It's the question at the heart of bioethics. And for a long time, the law has generally sided with the researchers and institutions. There was a famous case in 1990, Moore v. Regents of the University of California.
Okay.
It wasn't about the Lacks family specifically, but it set the precedent. A man named John Moore had his spleen removed as part of his leukemia treatment. His doctor, without telling him, patented a cell line from his spleen called the Mo line.
Of course he did. And Moore sued.
He did. And the California Supreme Court ruled that once tissues are removed from your body, you no longer have property rights to them. They are essentially considered biological waste.
That seems wild. I mean, it's my body. It came from me.
The court made a utilitarian argument. They argued that if every patient owned their discarded tissues, medical research would grind to a halt because you'd have to track ownership for every single sample in a lab.
Okay, I see the logic, but it still feels wrong. However, the Lacks family didn't give up. There was a much more recent lawsuit, right? Against Thermo Fisher.
Yes, very recent. In 2021, the estate of Henrietta Lacks sued Thermo Fisher Scientific,
And they're a huge company.
a biotech giant that mass produces and sells HeLa cells by the vial. The estate argued that the company was unjustly enriched by profiting from what was essentially stolen property, the cells taken without any consent.
And what happened with that case?
They settled. In August 2023, on what would have been Henrietta's 103rd birthday, they reached a settlement for undisclosed terms.
So we don't know the amount.
We don't. But the fact that a massive corporation like Thermo Fisher settled rather than fighting it all the way to the Supreme Court suggests a real shift in the landscape. Companies are realizing that the standard practice of 1951 is a major legal and PR liability today.
I'm glad they got something, finally. But let's talk about the recognition that isn't monetary, because for so long she was just this ghost in the machine.
The cultural shift is largely, and I think rightly, credited to Rebecca Skloot's 2010 book, The Immortal Life of Henrietta Lacks. It was a massive bestseller.
It put a human face on the story.
It did. Skloot worked directly with Henrietta's daughter, Deborah, who was just desperate to know the mother she barely remembered. The book didn't just tell the science. It told the story of the woman and her family.
And that book really kicked off a wave of acknowledgement.
A huge wave. There was the HBO movie with Oprah Winfrey playing Deborah, and then the memorial started. There are statues now. The University of Bristol in the UK put up a statue of her, which is actually the first public statue of a black woman created by a black woman in the UK.
That's very cool.
Johns Hopkins, the very institution where this all started, named a research building after her in 2018. The WHO gave her a posthumous award in 2021. And perhaps most touching of all, her grave finally got a headstone.
Tell me about the headstone.
In 2010, Dr. Roland Patillo, a scientist who had actually been mentored by George Gay but was also a fierce advocate for the family, he donated a headstone. It's shaped like a book. And the epitaph reads, "Here lies Henrietta Lacks, HeLa. Her immortal cells will continue to help mankind forever.
That's beautiful. It acknowledges both sides of it. And they did one for Elsie too, didn't they?
They did. Using some of the proceeds for the book, the family was finally able to mark the grave of the daughter who was lost to that asylum. It brought a sense of closure, or at least a dignity, that had been missing for over 60 years.
So, as we wrap this all up, we're left with this incredible duality. On one hand, you have medical salvation, the polio vaccine, cancer treatments, HIV research, literally millions, maybe billions of lives saved.
Mhm.
I mean, if you are listening to this right now, there is a very, very high chance that HeLa cells have impacted your health or the health of someone you love in a positive way.
Absolutely. The scientific utility is undeniable. We can't erase the good that came from the cells, but at the same time, we cannot erase the human cost.
Right.
The exploitation of a poor black woman in a segregated medical system, the decades of keeping her family in the dark, the poverty they lived in while corporations profited from their mother's biology.
It's one of those stories where two seemingly contradictory things are true at once. The science is a miracle, and the origin is a profound violation.
And that's why we have to tell the whole story of Henrietta Lacks. Not just to praise the cell line, but to honor the woman.
So here is a thought to leave you with, and it's a tricky one. We live in the age of 23andMe and Ancestry.com. We are constantly swabbing our cheeks and sending our biological data out into the cloud.
We are. All the time.
The Supreme Court precedent says you don't own your discarded tissue. But as our genetic data becomes more valuable than oil, do we need to fundamentally rethink that? If you go in for a surgery tomorrow and your waste tissue holds the key to curing Alzheimer's and a pharma company makes $100 billion from it, do you deserve a cut?
Or does your biology belong to humanity the moment it leaves your body?
It's the central question of the next century of medicine. Just picture it. The cells in that roller drum Dr. Gay built, turning once every hour. Still dividing today, more than 70 years after Henrietta Lacks took her last breath.
She's still here?
She is the immortal Henrietta Lacks. Thanks for listening to this deep dive.
We'll catch you on the next one.